Hey everybody, this is Mr. K coming to you from the Herberg Science Laboratory with another remote lab video. So in this remote lab we will be detecting cellular respiration. Cellular respiration is a cell process that gets the energy out of glucose. It releases carbon dioxide gas as a product. Now let's take a look at some of the materials for this lab here. We have test tubes. I've labeled them A, B, and C. This may be the only thing that you won't be able to get at home. You may be able to find an alternative to the test tubes. We have balloons. We're going to have 3 balloons. 1, 2, 3. A couple of packets of sugar. And some dry yeast, active yeast. It's the stuff that you use to make bread. You can get this at the supermarket. You're going to need some warm water for the yeast solution, about 2/3 cup for every 1 packet of yeast. And 1 packet would be enough. If you don't have a packet of yeast, it's about 2 1/4 teaspoons. And you'll need something to mix it up in to make your solution. Those are all the materials. So let's talk about how we're going to get it set up. Okay. Got our test tubes labeled A, B, and C. Next thing we want to do is we're going to create our yeast solution. So you'll get a mixing bowl or something like that, about 2/3 cup of warm water. Okay, yeast, and if you can see it in here, it looks, maybe you can't see very well, it looks kind of like little grains of sand. It's very, very tiny. And each of these is a single-celled organism. It's a fungus, so it does cellular respiration in order to get energy out of sugar. So we add the yeast in there. And then once we've got the yeast and the warm water together, get a mixer, stir it up until your solution is very, very well mixed. Okay, good enough. Now, I have found it easier to pour it into the solution by first transferring it into a beaker. Maybe that'll work. But you're going to end up with about this much solution. This is definitely more than you would need in one setup. Okay, and you can't smell it, but it smells kind of stinky. And so, what we're going to do next is we're going to add this yeast solution to the 3 test tubes. And we want to keep it so that we've got about the same amount in each one, as best as we can. And then by the end of it, you'll probably end up with about, maybe about half of the way filled up. A little bit more. A little bit more. So you notice they're all about even. And it's a little bit more than halfway. You could be a little bit less than halfway. Doesn't matter too much. Okay. Okay, next step is going to be add our sugar. Now this is a controlled experiment. So we have a manipulated variable. What we're changing the amount of sugar. So in C, we're not going to put any sugar at all. So we can see what happens if there's no sugar in the solution. In B, actually, we'll do it. We'll take 1 pack of sugar, and we're going to put about half of that pack of sugar in A. About half of it. I'm going to estimate a little bit. Okay, and then we put the remaining amount of that packet into B. Okay, and we're going to take one more packet of sugar. Okay, and in this last packet, we're going to put all of it in B. So what we're going to end up with is A will have a little bit of sugar, B will have a lot of sugar, C will have no sugar. And that is so that we can compare the different amounts of sugar to see which one makes the most difference in cellular respiration. Okay? Now, your next step is with these balloons here. The smaller the balloon you use, the more you're going to be able to observe gas being trapped. If the balloon is really big, you might not actually see it inflate at all. If the balloon is really small, it's more likely you will see something happening. Okay, so got it all set up. Now what we're going to do is we're going to put the balloons on top. Okay, there's one. To do it easier, I like to kind of move these around like this to stretch it out a little bit. Make it easier. Two, and then the third one. Okay, looking good. So one other thing we want to do before we start doing our observations is you want to just mix it a little bit, but to mix it, the best way to do it would be, imagine you're drawing a little circle on the ground, or a bigger circle on the ground, so you can see how I'm doing it here. So we're going to mix it up, make sure it's nice and mixed, but we don't want to get any of the solution like in the balloon or anything like that. Okay. All right. Now we wait and watch. So what you're going to do is you're going to observe what's happening here. You're going to watch carefully to see which ones begin to bubble. That's the carbon dioxide coming out of the solution. It'll begin to foam, it'll look different, and you're also going to monitor the balloons, because as the tubes fill up with carbon dioxide gas, the balloons will begin to inflate, and you'll be able to see a difference there. You can squeeze the balloons to see which ones have more or less air in them. But that's the setup. So we're going to end this video here, and I'm going to start the next video up in a moment. The next video will be just the camera on the setup. You're going to watch the camera nice and carefully and make your observations, one observation every minute. You'll see on your data table that there is a section in the data table every minute for you to answer a question or highlight what you've observed.